thank you to everyone for joining us today. I'm John Yoder with Axiom Healthcare Strategies. The focus of today's town hall is going to be around radioligand therapies. We have the pleasure of with us today of joining Dr. Sartor as well as Dr. Tagawa. We'll dive into some of the latest and exciting developments that we've seen within radioligand therapies and what they might unfold for that of oncology. But I will pass it off to each of them to give a quick background on themselves, and then we'll dive in.
Hi, well, I'll go ahead and take the lead. I'm Oliver Sartor. I'm a medical oncologist, long interested in the theranostic field, focusing on prostate, and delighted to be here today.
A very similar answer, except a name difference. Scott Tagawa, I'm a medical oncologist. I do see and treat all of the G malignancies, although prostate cancer is the most common that I see. And like Oliver, interested in multiple different developmental therapeutics and biomarkers, but have sub-focused on cell surface targeting, and much of that has been with targeted radionuclides.
Excellent. Well, thank you both. I'm looking forward to today's discussion. Recently we've seen a lot of, I would say, deal activity, particularly within the radioligand therapy space. Whether going back to Point and Lilly, Raise Bio with BMS, as well as kind of most recently Fusion with AZ. But it'd be kind of interesting to almost start at what you can maybe call a pivotal point around the radioligand therapy space with the approval of Pluvicto, and thinking about how has kind of the entry of Pluvicto started shaping and impacting the use of radioligand therapies today within prostate cancer.
Scott, I'll jump in. I'll do first on this one. Next time you do first. So we'll alternate. I think Pluvicto was an amazingly important move for the field. If we go back in time, I think that radium was catalytic because it really did prolong survival in a prospective randomized trial. But I think there were some issues around radium that led to some disappointment, including the lack of PSA decline and sort of the lack of positive follow-up data after that initial Simca trial. So some of the enthusiasm may have been on the wane, and the category that is an exception to that was the neuroendocrine tumors, where clearly lutetium began to be important, very important. Mm-hmm. But the problem with the neuroendocrine tumors is they're small in number, very important if you have it, but from a larger perspective, more population-based, neuroendocrine tumors don't really change the landscape. Typically, the specialized centers will take care of these patients, and they don't really penetrate that much into the day-to-day life. Well, I think Pluvicto changed that. First of all, he had a bit of a buildup prior to the Vision trial with the data coming from Germany. And if I may say so, the PSMA antibody data coming out of Cornell with Scott. And so the anticipation was kind of rising. And then now the positive trial disseminates broadly, I think, across the oncologic community. Patients know about it. They want to receive it. And I think it's a little bit of a catalytic change as compared to the neuroendocrine, and a bit of a catalytic change in comparison to the radium. So I think the impact of the Vision trial is large, and you see, I think, a lot of this deal-making that has occurred in part because of the enthusiasm in the field. I think in part that was fueled by the Vision trial of Pluvicto. Now, that may be a little bit of a overly focused view of the world because there's so many other things you could take into account. But I don't know, Scott, would you agree, disagree, or counterpoint, or expand?
Yeah, maybe. So agree and maybe expand a little bit. So I was going to say very similar things, I guess not so surprisingly. But if we think, kind of bigger picture of theranostics, so you take, iodine for thyroid cancer as the prototypical theranostic, but a small disease. But that was kind of proof of principle, and the CD20 for immunotherapy wasn't, I don't think, the right time, and had some competing treatments that the physicians could give and make money on, the different types of things. And then we have two approved drugs, and I think they're both great for prostate cancer with overall survival benefits, not a lot of toxicity, but no responses that we can see. And they don't sell so well in terms of CYP and radium, even though I think they both have a place. And then we have exactly what Oliver's saying, something that is well tolerated, improves OS, progression, responses, quality of life, etc. And that's with my viewpoint, a tolerable therapy, not no toxicity, but a tolerable therapy with even with the higher number of AEs in the experimental arms, at least in vision, with higher grade AEs actually in the control arm of PSMA4. But in any case, then you're also getting a disease that pharma is paying attention to, numbers in terms of prostate cancer. Patients are seeing responses. It's always nice to see, PSA. I spend half my day telling people to don't worry about PSA so much in the clinic, but we focus on it and patients focus on it.
Yeah.
And then the additional kind of buildup, I agree with the buildup over a long time. The additional buildup was imaging. So, whether it was where's my PSA coming from, or initial staging, whatever it was, imaging became available. And we're not talking about, Caprimab, Indium-111, Prostate. And we're talking about real imaging that actually worked, that became the standard of care in many places, but was not available to most Americans unless they traveled or they participated in a study. And that was a big buildup. So, and patients, all these pictures online or conferences, or like, oh, wow, I can see all these things. And that all, I think, came together at around the same time. So I think that's why, there's so much in the field right now. Oliver longer than me, but Oliver have been plugging along, and now there's just a ton of stuff, which is a lot of fun for us.
Yeah, it's kind of, we almost hit that tipping point where I think to each of your points, like we have the imaging, right? And I think one of the interesting points that you brought up as well, Dr. Sartor, is that within the vision study, they were able to demonstrate kind of the change in PSA, right? So I think from an adoption perspective or patient understanding, or maybe your more broader perspective, clinician, it's something that they can more relate to. Obviously, overall survival is critical. But I wonder, as you guys think about, vision starting in the later line setting where it is today within MCRPC, where obviously PSMA4, SPLASH, and others, we're starting to see that shift towards earlier stages within prostate cancer. I'm curious to hear your perspectives on the role, and this could even go beyond, lutetium-based agents. I'm thinking about what is that role of a radioligand therapy within earlier stage settings within prostate cancer?
So, in terms of drug development and oncology, this is, and even outside of prostate cancer, this is typically it. What's the biggest unmet need when there's nothing else? Easier to go against nothing or weak compares, get an approval, and then move forward. That's kind of been what's happened in terms of drug development kind of forever. There's also, safety issues. When it's more unknown, it's safety. So it makes kind of sense, and it's not surprising that it's happening. Scientifically, I would say, if we stick just with what we have, scientifically, it makes sense that it should work much better, right? There's less risk of not having the target there. Oliver mentioned neuroendocrine tumors in terms of the setting of somatostatin, but neuroendocrine prostate cancer, loss of AR, loss of PSMA, is a lot less common in earlier line settings. Some of the radiation resistance, particularly, I think, more likely impact for the betas and the alphas. We'll get to that later. But those radio resistance, or just actually treatment resistant, because p53 is maybe universal for a lot of different drugs, but there's less of that earlier. So absolutely, and if you're going to combine with something which is typically AR, where there may be PSA modulation plus radio sensitivity, that all absolutely makes sense to move forward. And then in a way, this is not exactly what you're asking, but in a way, we're using, quote, radioligand in terms of the imaging perspective right now to do treatment. Kind of even in the absence of major long-term data, we see something, we zap it or do something with it. So it kind of is already there. I think we all acknowledge that there must be something that we're missing. PSA is going up, we see one thing, and it's nice that we have a much more sensitive imaging agent. We did it with bone scans in the past, but now we're capturing a bigger percentage, but it's prominent everything. But a molecular agent that does the same thing, even if we can't see it, might be able to land in these smaller lesions and treat them. I think that is the next wave. And each step of the way, moving earlier, we do accumulate long-term follow-up. So the safety pool of data is also increasing, even though, I mean, I have a handful of patients that have 10-year follow-up, but it's a pretty small number.
Yeah.
Yeah, so I like Scott's answer, and I think it's a nice interplay which he introduced as well between sort of the imaging side and therapeutic side and how they merge together. Very critically, we do think about the diagnostic side turning into therapeutics with the utilization of targeted radiation, and that is an absolute part of our standard of care today. We look for these small lesions. We certainly know that there could be a multiplicity of more lesions that we're not yet visible, not yet seen on the PSMA PET. But nevertheless, by taking action against some of these visible lesions, we can see some positive effects in a certain proportion of patients. So that we can see it, we can treat it is more than just the use of radioligand therapies. It can also be the imaging and the external beam and more. So that's a really good point. Secondly, just to amplify and expand a little bit on Scott, we have started to look at these bigger trials like PSMA-4, like the SPLASH trial, that are beginning to report data. There's an important data update just last week from Novartis. I suspect many of you may have seen it. The original PSMA-4 data had really outstanding effects on rPFS, time to progression, overall response rate, tumor shrinkage measured by objective radiographic criteria, CAT scans, etc., PSA, health-related quality of life, and adverse events look better. You kind of go down the line, and there are a series of positive effects. However, in the intent to treat, which was an important analysis done for overall survival, there was no benefit. Now, importantly, 84% of the patients crossed over from the control arm to the Pluvicto arm, and I think that colors the outcome. But nevertheless, if your hazard ratio is above 1, meaning the patients randomized to the radioisotopic therapy actually did a little worse when it came to survival, then there's a pretty big distinction between over 1 and less than 1. And I know that confidence intervals are still going to be overlapping, but nevertheless, the recent press release is okay, now the hazard ratio is actually less than 1 as opposed to more than 1. And it's not a wholesale change, but I think it does give a little more probability to a favorable response from the regulators, the FDA in particular. Of course, we have to wait and see, and I'm very respectful of the regulators, by the way. They've got a tough job, and I certainly don't like to criticize the FDA. They've got a massive job that they have to perform. But I think they'll take a careful look at the data. And what I'm hoping is sort of totality of the positive data from the rPFS to the PSA to the ORR to the health-related quality of life, the adverse event profile taken in ensemble will create the positive scenario necessary for approval. Now, just a brief caveat, I don't want to talk too much because I know we have a lot of other topics, but as you do move earlier in the course of disease, it means that the patient is going to be living longer after treatment. And there is some concern, and I think some reasonable concern, that longer follow-up may translate into the possibility of delayed toxicity. I didn't say the inevitability, but the possibility. And people who've been radiated may have effects that emerge later in their treatment course. And I think we're all very vigilant on that possibility. So far, we really haven't seen very much, at least with lutetium. In fact, it's not with radium either. But we need to watch for it, and we need to have a cautionary message about bringing isotopes earlier, because we don't really know the long-term side effects until we observe them. And so far, they're not really observable. So probably too long a comment, but forgive me.
No, I mean, it's a great point, right? As you think about moving any therapy earlier, particularly in its early days of clinical development, understanding what those long-term implications are going to be challenging. A little bit of a transition in thinking about, right? So, because I have this thought that I'm trying to understand the core difference between alpha and beta emitters. And where I'm going with this, with the moving earlier, is I'm wondering if there is some kind of perspectives that you might have around their role within, one, the differences between the two. And then secondly, if one moves earlier, is there a sequencing consideration that we need to be thinking about as new, more kind of types of radiotherapies come onto market?
I'll start, and then I'll try to be brief, and maybe Scott and I'll go back and forth. That's a big and complex question.
It's a big one, yeah.
Yeah, no, it really is. So, alpha betas, first of all, the particle differences, I think, are evident to most people on the call, but you got the difference between 2 protons, 2 neutrons, versus some little flying around beta particle. And it's a little bit like a battleship and a little speedboat. There's a big difference here in the power of the particle. But in addition to that, on the biology, you end up with more double strand breaks with the alpha, and a very short, extremely short area of deposition once the alpha particle has been deposited on the cell surface. It doesn't travel very far, typically somewhere between 40 to 80 microns, where we're talking about maybe a millimeter or so for the betas. Bottom line, I think overall, if I had to just make a big statement, if I were a cancer, I'd be more afraid of an alpha than I would be a beta, because I don't want that big wrecking ball going through my DNA and messing up my future. And I think the alpha particles is pretty damn good about killing things, and the beta particles good, but it requires crossfire and a little more effect. So I think, head to head, if you get the dosing right, and assuming the toxicity is okay, I'll take an alpha over beta. And if you talk about sequencing, we have very clear data at this point from a variety of the German retrospective studies and others, India, Turkey, Chile, etc., that alphas plus beta can, to some extent, be a successful sequence, that you can overcome the beta resistance. On the other hand, have very little data that would suggest that betas can overcome alphas. I'll stop there. And like I said, such a big question. I don't know. I want to hear Scott.
Yeah, well, I'm glad you got to the inverse of that, right? Because obviously, I'm a little more familiar with the alpha post beta, but I was wondering if you do the inverse of that, right? If you have a beta post alpha, do you see this a similar effect?
Well, I think we have a lot of unknown, and so the facts are we don't have a lot of data there.
Right.
I don't know. Let's hear your thoughts.
Yeah, so my high level view in terms of alpha versus beta, as I talk to patients or whatever else, I would say, an alpha in the right place, i.e. cancer cell, I'll take it over beta. In the wrong place, maybe I'll take a beta. So it kind of depends. That being said, if whatever the cell is, whether it's a parotid cell or something else, an alpha is in it, it probably is going to die. So I'd say in theory, maybe there's less risk of future malignancies with an alpha. I don't know that to actually be true, but that could be a hypothesis. We have some data of betas after alphas. So the level 1 data is just different targets. So many of the prospective studies, including Vision, they had at least hydroxyapatite targeted alphas, meaning they had radium first. So we know that in terms of betas after alphas, there can be some efficacy. There's anecdotal data that at least I have personally, that they happen to be on an alpha trial first, and then later, get commercial beta or a different experimental. So we know it can work, but I agree with Oliver that in terms of PSMA targeting, the biggest majority of the data, both prospective and retrospective, is alpha following beta, and clearly there is a real response rate like that. But remember, there's also a response rate to retreatment with beta. So you don't say no, it's kind of different. So in any case, I think that's the bigger direction. If we talk about the same target with PSMA, I think it is alpha after beta. And just think of the trials and the new drugs that are out there. It doesn't kind of matter what something starts at today, whether it's alpha beta, but many of them are alphas. Because of the landscape today, as well as the predicted future landscape, it does make sense they're also positioning themselves after betas because they're already there. And it's going to be
there, right?
Particularly if trials like PSMA addition are positive, then there's going to be just a lot of patients that are getting it pre-CRPC.
Got it. No, that's super helpful. Is there? Obviously, we have if I'm a cancer cell, I fear an alpha much more. What are some of the differences between the two clinically from the patient perspective, right? So if I were a patient receiving either one, what would I expect from an adverse event mindset?
I think the target is a little bit more important than the alpha versus the beta in terms of just the list of what might happen. That being said, what actually happens in that list may be a little bit different if it's an alpha versus beta. So for instance, if we're targeting the PSMA, probably any systemic radiation or even external beam radiation might have some constitutional things like fatigue or something like that. But if we're targeting PSMA, whatever it is, something that's going to hit the target that is off tumor, meaning parotids or something like that, I would say, dry mouth is going to be a risk of alpha versus beta. But because of what I said before, in the wrong place, it's going to be more deadly. Then maybe it's going to be higher grade and longer lasting. The reason I'm saying maybe is because we don't have any comparative head-to-head data, but it seemed like that's the way it makes biological sense that an electron damages, but the proton neutrons kill. Basically saying the same thing that Oliver said in a slightly different way, that it may be more potent both against the cancer and against whatever organ it happens to land in. Luckily, whether it's retrospective or prospective, there hasn't been a whole lot of major bad things that have been reported. So let's say xerostomia and grade 2, I think can affect quality of life. But even but grade 3, which is we don't really have reports of that, which is great because that means they can't eat anything. Even with the alphas after the betas, it still appears to not being happening. I think part of that is because, in the retrospective world, Germany, South Africa, wherever else is, they have experience and they're not going to push it. And then prospectively, we're being careful. Might it happen? Sure. It's all, an efficacy toxicity ratio in terms of what we'd expect. But in any case, that's where I'd say the list is probably very similar. I'm trying to think of something that would be totally different, like not happen with the beta. I think it's basically the same categories, just the severity may be a little bit different. And maybe actually something like bone marrow might be worse with the beta just because it kind of depends on obviously where it lands. But the circulation is probably less important for the alpha because of the short range. Landing has to be right next to a stem cell to have that kind of off target effect, versus the beta can be further away and still affect the bone marrow. So I'd say at equivalent kind of radioactivity doses, I think an alpha probably is a little less in terms of that, but we don't give the same equivalent radioactivity doses. So it's not exactly that issue.
Yeah, it's interesting. You actually brought up something that I was going to push on later, but that's perfect. I wanted to understand some of the kind of the bone marrow toxicity concerns. As you start thinking about sequencing or eventually combinations with RLTs, right? So, particularly from a beta emitter perspective, is there concern that some of the bone marrow toxicities could potentially then preclude a patient from receiving other treatments such as chemotherapy later on? And how might you weigh that of alpha versus beta? Obviously, understanding a lot of the data to date, it might be too premature to make a definitive decision.
Yeah, I think it's a good question, and we don't really know, and so I think we have to be very careful about making conclusions.
Right.
As it turns out, Scott mentioned that target matters, and we're now talking about isotope matters. But remember, there's another thing that can matter too, and that's the ability to carry the molecule into the target with isotope attached may be in part dependent on the antibody versus a small molecule versus something in between. And as we're looking at sort of marrow toxicity in particular, it looks like the antibodies with a longer circulation time may end up with a little more marrow toxicity. But now you have to realize that that'll be counterbalanced by the less activity you're going to see, say, in the carotids, and less probability of renal toxicity because you don't have the rapid renal excretion. So there's going to be a little bit of a yin and a yang. One of the things I think that's a little bit interesting is that there are now molecules under development that are neither antibodies nor the typical small molecules. I might mention those that are albumin binders, for instance. There's some that are kind of tunable albumin binders that it's a longer half-life than, say, the 617 or the INT small molecules, but a shorter half-life than the antibodies. And then also some antibody engineering. Telix, for instance, has got a 592 that they call it, taken from Scott and Cornell's 591 and then engineered for a little more rapid disposition. So as we go forward, I think we have to take into account the target, the isotope, but also the delivery vehicle, which can determine important parameters that are going to be associated with different toxicities, even the same target and the same isotope. So that's, I think, something important to keep in consideration. I'll probably go on, but I'll stop, because I try to make sure we're focused here.
Yeah. No, much appreciated. And maybe if we add one other aspect here, you were mentioning the DNA breaks and the ability that both alpha and beta particles would have, and potentially resulting in neoantigen release. So I'm curious if you then think about, to John's earlier question around sequencing, are there particular combinations? Typically, when we think of progression on one line of therapy, if you look for sequencing, you might think about a combination that you're excited about, maybe IO or other mechanisms of action. Open question.
So I guess that was a yes, no, so answer yes.
Yes. Okay.
And I'll expand on it. So, I mentioned this before, but, the most natural one is AR, and that was actually most of Vision. So you have to remember that Vision was actually a combination study. And it's nice to have the, PSMA4, which showed the single agent data, but so in most of that combination in terms of the protocol specified standard of care turned out to be AR targeted therapy. So it makes sense because there is data that, at least initially, we're going to upregulate PSMA, so the target expression will be higher. And then we also know that AR targeting can lead to radiosensitization. So it's like a double-pronged reason that may help the radionuclide. And then AR targeting just may work on its own. So, that's the idea in terms of PSMA addition, for instance. So I think we may get a bigger bang for our buck with that particular approach. There's other ones that will, also make sense. So, and many of these have already been done. So, LoopARP was presented, and I think there's a couple of different ways of looking at ARP inhibition as well as a targeted radionuclide. One of them is to act as a radiosensitizer, and that's the way the LUPARP was done, not picking out tumors or patients that are necessarily predisposed to have a good response to a PARP inhibitor, not giving full doses, but figuring out a dose that would be safe and giving it only around the time of the lutetium, which should, the idea is you damage some DNA, single strand DNA, right? So you disallow the repair with, in this case, olaparib, but any PARP inhibitor may kind of work in theory, and not randomized data yet, but that is another approach that makes sense. Forgetting which radiosensitizer, but Louis Emmett has a publication on lutetium PSMA with a radiosensitizer as a kind of fully published study. Chemotherapy, in a way a little bit similarly, but a little bit different in terms of AR targeting. So chemotherapy can work on its own, but taxanes also have been shown to radiosensitize, and taxanes may hit a different compartment, so a PSMA negative or low kind of compartment. So that makes sense. There's the lutetium safety with the antibody JF1, and lutetium JF1 with docetaxel, and now there's ongoing prospective study with cabazitaxel, so that does make sense. I would say that my view of radiation with immune checkpoint inhibition is that it's fairly clear that we cure some mice with that approach. Hard to know how much that works. Most of it has been with external beam, and maybe there's some abscopal effect. A difference using a more of a systemic approach is it's not just at one, maybe we're able to, first of all, just hit many more targets and it works on its own. But then also, if there's a quote needed abscopal, then it's maybe just within the tumor or other areas that are target low if it's PSMA. So 2 out of the 3 studies that have been, one's published and 2 have been presented, are from in my mind, the immunologic approach of saying, okay, what can we do to make the PD-1 work better? And that's with a single dose of lutetium PSMA or a single dose of actinium PSMA, versus, this is my view of Prince Australian approaches, taking the radioligand as the standard and adding, kind of full dose of both, adding pembrolizumab to that. And, it clearly is safe, and we'll have to see what happens in terms of efficacy from a long-term trial. But anyway, those are the different targets that have already been used in the clinic. I guess we don't really have randomized data of something being added to the PSMA targeted radionuclide, but obviously adding something to that, i.e. vision, that does work, or in phase 2, enzalutamide.
That's a really thorough and good response from Scott. I don't have much to add, honestly. I would say that when we think about DNA repair, I do think beyond PARP in a conceptual way. We have PARP inhibition now in combination, and they're provocative data. I won't say that they're definitive, but Shanine Sandhu at Peter Mac, I thought presented some pretty good data. And if we look at some of the animal models, in vitro models, it could be that PARP inhibition is not the ideal inhibitor of DNA repair. There might be others that are better. So keep your eye out on that. In terms of the various radiosensitizers, I agree completely with Scott. We have some data with AR already, and that's a really good basis for going forward. But there's 2 separate issues in my mind. There's the sensitization that I think is really important, but there's also the potential for clonal heterogeneity that could be important, particularly with some of the chemotherapies. All these cells, even the ones that look like they're fairly hot on a PSMA PET scan, doesn't mean that every PSMA is going to be expressed on every tumor cell. You are going to have some heterogeneity, and if you end up killing more than one type of cell with your different therapies, then that might be also a productive way to approach the cancer. So synergy, sensitization, yes, but also overcoming heterogeneity, yes. I think we're at the infancy. If you were to draw back and just briefly say, well, let's talk about combination therapy in oncology, it's a gigantic topic. And when you talk about the radioisotopic therapies and combinations, it's also a gigantic topic. And I think it is logical for us to conclude that a variety of combinatorial therapies are going to be tested, and I suspect that some of them are going to be pretty good going forward. But we had to put a sort of stake in the ground to show the radioligands made a difference before we can start going to those combinations. But I think the time to look at combinations is now. Let's not hold back. There's so damn much data now that we can look at, and Scott's been very active. Scott's done immunotherapy in combination. Obviously, we've all done hormonal therapies. Scott, you've done, I know, some interesting work even with alpha beta. We're talking about combinations of isotopes. So there's so much to do.
And just that's one thing I forgot to even mention, kind of our own data. Alpha betas, it absolutely makes sense. You talk to the physicists and you're going to kill more cells. But even just targeting, and we haven't done it so much, but just targeting the same cell with antibody and small molecule. So there may be some synergy that is there, regardless of what is attached to either one of them.
Mm-hmm. Good point, Scott. I agree.
Makes perfect sense. We have a live question from the audience, and I think this might be helpful for just general understanding of, when a patient is selected for radiotherapy, what does that kind of full cycle look like for them? And maybe we kind of think of this while we've been talking a little future facing from a kind of combination perspective. What does a standard treatment look like today for a patient who's going to come in and receive Pluvicto?
I'll give kind of a quick answer. The first thing is that we're treating either in two contexts, either on label or in a clinical trial. I'll take that as an on-label question because the clinical trials get a little complex. So first of all, we want to understand their prior therapies, and today that means an ARPI such as abiraterone and enzalutamide with progression thereafter, and also a taxane and progression thereafter. Obviously, ADT is part of the standard, so people need to progress after a series of therapies, not just one, unless you're using the triplet therapy up front. That's a minority of patients. Then we want to make sure that their blood counts are adequate. Somebody comes in with a thrombocytopenia platelet count of 15,000, that's not a good start. So we'll run a quick survey on their basic blood counts, chemistries, etc. And then, of course, we want to understand, do they have PSMA PET uptake in a manner that we believe will be commensurate with targeting with the Pluvicto type agent. So we run a PET scan, and that PET scan is going to be looked at from several perspectives, not only the distribution of metastatic disease, but the degree of uptake. And in the VISION trial, we had a cutoff of liver. We kind of look at the liver, and then we say, make sure the metastatic disease is coming in with an uptake greater than liver. And then we're also looking for these PSMA PET negative lesions. And here we're going to simply define it as PSMA PET uptake less than liver. And if we see, for instance, the liver has got a whole bunch of lesions and they're cold on PSMA, that's a bad reason to treat that patient with Pluvicto. So bottom line is, prior therapies count. The ability of the PET scan to be able to exclude and include patients that are appropriate, that counts. And all the chemistries and evaluations, prior therapies, scans together comprise the evidence that we need to proceed. Now, if the patient's ready to go, we say, order it up, we're ready to go. Typically comes out in, somewhere between a week or two later. Infusion short, then we set the monitoring. That hopefully that answers the question that came in. If not, I can elaborate, but I think that's a pretty good picture about who we evaluate and kind of what happens in the process.
Yeah, no, that was helpful, even though like as a non-clinician, you read it so many times that you think you understand, but I think hearing it from your perspective is very helpful.
And I think as we're discussing sort of the operational aspect of radioligand therapy, it's important to remember that there, and Dr. Tagawa mentioned this earlier, sort of the specialized, this is a specialized field, but a growing one. And I'm curious also, there's been a lot of discussion around how important dosing is, how important it is to get dosing right. And with your respective expertise and experience in designing clinical trials, would love to hear your thoughts with regards to how to improve the study of dosing for radioligand therapy in the future. Is it still 3+3, like dose escalation and expansion? Are there any creative ways we can think about designing trials to make sure that we're addressing the dosing question properly for the future?
So let me just set the stage, and I think answer your question in terms of the way you're using the word dose. But so to me, to make it as simple as possible in terms of how much success we can have, it's dose and radiosensitivity. And what I mean by dose is how much radiation is hitting that cell. And there's different aspects of it. One is how much target is there. Another aspect is how much gets there and stays there. Another aspect is how much do you squirt in or infusion. And that's what I think you mean by dose. But anyway, those are the different things. But I was going to say, as a radiation oncologist, I'm not a radiation oncologist. From a radiation oncology or for physics perspective, it is what is there. And a certain patient, you have whatever's in the syringe and give it to one exact same in a different syringe, it's going to be a different dose to tumor based on those other aspects. And then the additional aspect of the outcome is going to be the radiosensitivity. So anyway, but I just want to kind of clarify that out there. So I think that now answering your question as my viewpoints, and I am a medical oncologist, so is to develop these as drugs. Now, there are so many permutations. I don't think that every single one can be figured out, but I think the two questions are how much is in the syringe in terms of how much radioactivity, and then when is the next squirt in going to be, meaning how the schedule part. So those are, I think, the two biggest ones. I do think that there should be some sort of dose escalation in terms of at least testing different amounts in the syringe or the bag, depending on how it's going in. That I do think it's worth it, both from a efficacy as well as a toxicity point of view, and something that is different that we actually have with virtually all of these, and these do include things like alphas, like actinium, is that we can also do dosimetry, meaning we actually could see where these land and how long they stay there. So some of them are harder than others because there's short-lived daughters that don't have a lot of energy, and it takes technology to be able to look. But we can, in sophisticated centers that are doing these phase 1 trials, we can look at that both in the good and the bad, meaning how much is in the normal organs, how much is in the tumor. And I think what we want to do, it's like having pharmacokinetics and pharmacodynamics in the traditional sense. This is something we can also see that's there. So my answering, research or trial design, I think that is right to have in early phase clinical trials to then figure out what is going to be the best going on to phase 3. Now, I think we can do the opposite. So I think that there are probably, this is my bias, more optimal ways to give lutetium PM617 than 200 millicuries every 6 weeks. We dose escalated and did just an entire course in 2 weeks. And I think that may be as good or better, but so I think it could be done in the other way, but then it's much harder, as an investigator, because pharma's already set, they're going to do it. It's harder to get that. And then we want to give higher radioactivity amounts, do it on our own or ask them to ship. There's a lot more complexities with doing it after the fact with the target radionuclide. So I think it should be done early, and dosimetry should be there. But I'll just point out from 2 weeks ago or so, the first publication from RPT Tech in JCO, Anna and George Skaros and others, to look at actually toxicity, like we're doing with drugs, as an endpoint or early phase clinical trials, but don't throw away the dosimetry either. So I don't think we want to use either one in isolation, but to have those two. And then I think that by the time we get to phase 3, that may not be the final optimization, but I think we're going to be a lot better off. And then the last point is just, we have to also keep in mind that it may be different in different clinical contexts, and certainly with combinations that may either radiosensitize or change the target or things like that, that the dosing schedule may be different.
I might add a couple things to what Scott said and agree with his overall appraisal. But, the FDA has now endorsed something called Project Optimus. Now, if you read Project Optimus, it's really interesting because in the documentation, it actually says that radiopharmaceuticals are excluded. But that's not really seemingly how they're approaching it. They broadly, and I think appropriately, want us to think beyond the older oncologic principles of the DLT, the dose limiting toxicity, and then back off one and give it. There could be a really good rationale not to give the maximum dose for these types of agents. And the Project Optimus is designed to create optimal dosing strategies before you get to the phase 3. So that's kind of point number one. I think we have to keep that in mind. The number 2 issue with sort of the FDA and regulatory matters that is really complex. Once a dose is established within a phase 3, you have the insurance companies and you have the standards of care that align behind that phase 3 dosing. And if you want to change it, you probably are going to need to change it in the context of a comparable study that looks at your dose versus the FDA approved dose. And guess what? That ain't easy. That's really hard. So once the dose becomes established and becomes a standard of care, you're going to enter into a completely different process for changing that dose. And I see, quite frankly, a lot of naivete, particularly among the nuclear medicine community. And they said, oh, but if we do dosimetry guided doses, it'll be better. And we do this. Well, guess what? Who's going to pay for that? If you want to fork out the $45,000 to do it on your patient, you're welcome to pay for it. But guess what? The insurance companies are not. And that creates a significant barrier to altering the doses that are established in the phase 3 and regulatory approved context. So bottom line is, I think we can do dosing better. I admire Scott for the work that he's done on dosing, even with PSMA 617 lutetium. And I don't think we're dosing people optimally today. I think we can do better. Now, imagine that you could be an up-and-coming little radiopharm company. You might be able to exploit this sort of poor dosing strategy that, well, I don't want to call it poor dosing. That's not a good word. Let's say suboptimal dosing strategy to your benefit, because I don't think Pluvicto is being dosed optimally today. And I think that there is an opportunity for improvement, even with the beta particle, given that the suboptimal dosing is going to be persistent, given the regulatory environment that we have today.
Yeah, you bring up an interesting point, right? And I think, obviously dosing, even when we think future facing, there's opportunities to do better, right? It's also a challenge based on some of the regulatory considerations that you laid out nicely. I'd be interested to hear a little bit around And we'll save the last few minutes for targets, because we've talked mainly about emitters for this full time, but around some of the current challenges that you guys have faced today, with, what are some of the barriers to adoption when it comes to radiopharmaceuticals?
So the one thing I was going to add to a couple of things, that when Oliver was just answering simple is not the right word, but the kind of practical thing about how do we go about that for patient. So the doctors that are the primary caregivers, not, I'm going to tell you they're primary care internists, but the primary caregivers in terms of making treatment decisions, medical oncologists and urologists, as a general rule, do not have licenses to be able to administer these. So it's a referral. Sometimes, I can tell you exactly, I'm on the third floor, one floor, literally, if I could cross through the floor, I'd be in nuclear medicine. It's, there. And other times it's miles away that they have to go. But that is something that is different on the patient side. And sometimes the patients say, do I really have to go meet someone I don't know? Sometimes they'll know the radiation oncologist, and sometimes they may know the nuclear medicine because they're getting it scanned, but it is a separate process. And then the separate part, which I think is a more minor issue, because I think all the doctors want what's best for the patients, but what is going into the pocket in terms of profit from chemotherapy versus going into someone else's pocket or a hospital, depending on what it is, for a radionuclide, at least in the kind of current landscape. So anyway, there are practical aspects, those two that are, a little bit different. And I'm actually forgetting your exact question, but the other part that we haven't talked about is, all of our anti-neoplastic agents, for the most part, we take precautions. We don't want whoever the pharmacist, the nurse, depends on what it is, if there's a hood, or don't touch it. Our take-home medicines, the ARPIs, we say, no one else should be handling it, particularly females, childbearing potential, et cetera, because it might go through the skin, but it's pretty easy with gloves, et cetera. This is a little bit different because depending on what it is, if there's a significant amount of gamma, like with lutetium, then they are exposing the public and others. So that's a little bit of a difference. Also, with these compared to some other agents, I don't think it is prohibited. The magnitude of the benefit versus that, as long as we counsel, in my mind, it's not a big deal. But Oliver and I've been doing this for decades now, so we're used to it, and that is at least a perceived barrier for a doctor, practice, or a patient who have never kind of been through that.
Yeah, I mean, briefly, and maybe a bit of a summary. So, the doctors who treat are not the doctors who care for the patient. That's a little bit of a barrier. You have to go from point A to point B. If I want to write docetaxel, I just write docetaxel. I don't have to make any referrals. And so that in and of itself is a little bit of a barrier. The financial barriers, I think, are smaller today, but in the older days, and I'll talk about the Bexar and Zeppelin, which were scientific successes and commercial failures, it turns out that these financial barriers to transfer from one specialty to another were, I think, larger then than now. But nevertheless, they still persist. And, if I don't want to be selfish in the medical sense, but I want to be realistic in the capitalistic sense, that the financials do matter. And to the extent that those financial issues can be mitigated by the way, in a large medical system, I don't get paid for the chemotherapy I give, and I doubt if Scott does either. And so I'm in a system at the Mayo Clinic that doesn't penalize me for the referral. But if you're in private practice, you could be financially penalized for the referral, and that's a barrier. There's also licensure barriers. The number of people who are comfortable with radioactive agents is relatively small. And I think some of the reimbursement side that goes to the provider administering these agents can be problematic. People need to be paid for their services, and they can't operate a charity when you're functioning in a medical system that dings you at every opportunity, whether it be the malpractice insurance you have to pay, or the overhead that you're going to encounter with your nurses and receptionists and more, you do have to make a financially viable decision when you're treating a patient. You just can't give those services away because it's going to come back. You got to pay for it somehow. Anyway, that may not be quite what you're looking for, but there's some very realistic barriers out there that I think need to be addressed and need to be talked about.
I mean, I think it's fantastic to hear, right? Because there's a lot of the real world implementation considerations that you guys face on a day-to-day that aren't kind of always considered. I do realize it's amazing how the hour has flown by. And we're definitely kind of want to be respectful of both of your time. I would happily invite both of you back. Hopefully Dr. Claes can join us next time. Unfortunately, he was unable to today. But it'd be great to hear some closing remarks from each of you and almost a little bit forward thinking. What are you kind of most excited about of what's coming in the near term? Obviously, there's a lot of experience today with PSMA. We didn't quite get to the targets discussion, but it was highlighted that's another kind of core consideration. So maybe what targets are you kind of potentially most excited about, and what's coming next from your perspective within this field?
So in terms of PSMA, I'm excited about the moving forward, different patient populations, combinations, and other drugs. Actually, even non-radionuclides. I think we have now validated PSMA as a target. In terms of additional targets, I think there's some that may be complementary to PSMA, meaning in a similar patient population that may be not entirely overlapping. And KLK2, I'd say, is the furthest along right now in terms of radionuclides, even though I'm waiting to see more data. But that being said, we think of overall in terms of cell surface targeting, we have a lot of validated targets across different tumor types for antibody conjugates. And I don't see why most of those couldn't also be used for targeted radionuclides. And some of those, if we go to the prostate space, are different than, only little overlapping in terms of PSMA. So neuroendocrine, for instance, DLL3, CEACAM5, et cetera. I think that those might have a role in the future.
Gosh, it's so damn hard to answer that question because I'm enthusiastic about so much.
Everything's good.
I think we're going to have better PSMA. I'm really looking forward to the alphas. I'm looking forward to different targets, HK2 being a good one, maybe, STEAP1, STEAP2 being others. I'm looking forward to the combinations. I'm just looking forward because right now it's extraordinarily exciting. There's almost more concepts than you can individually follow. And by the time you turn out, you think you've got it, you don't. And, I'm a little more enthusiastic about, say, lead-212 today than I used to be. I'm a little more enthusiastic about antibody pre-targeting than I used to be. And, check with me tomorrow. I'll probably have something else. It's a little crazy, but the field is so evolutionary right now. It's a blast to be a part of it, by the way. It's really been intellectually rewarding and personally rewarding to be a part of this space. And I'll just simply say, stay tuned. Things are going to change.
Yeah. Well, again, thank you so much. It's been an absolute pleasure having the two of you here today, and hopefully we look forward to more of these sessions in the future. So with that, I will wish everyone a happy weekend, and we will talk to everyone soon. Take care.
Thank you, John. Thank you, Scott. Enjoyed your discussion.
Great seeing you too.
Bye.